Section 2 of Gray's Anatomy, Part 3. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 3, by Henry Gray. The blood. The blood is an opaque, rather viscous fluid of a bright red or scarlet color when it flows from the arteries, of a dark red or purple color when it flows from the veins. It is salt to the taste and has a peculiar faint odor and an alkaline reaction. Its specific gravity is about 1.06, and its temperature is generally about 37 degrees, though varying slightly in different parts of the body. General composition of the blood. Blood consists of a fairly yellow fluid, the plasma or liquor sanguinis, in which are suspended numerous minute particles, the blood corpuscles, the majority of which are colored and give the blood its red tint. If a drop of blood be placed on a thin layer on a glass slide and examined under the microscope, a number of these corpuscles will be seen floating in the plasma. The blood corpuscles are of three kinds: 1. Colored corpuscles or erythrocytes, 2. Colorless corpuscles or leukocytes, and 3. Blood platelets. 1. Colored or red corpuscles, erythrocytes, when examined under the microscope, are seen to be circular discs, biconcave in profile. The disc has no nucleus, but in consequence of its biconcave shape, presents, according to the alterations of focus under an ordinary high power, a central part, sometimes bright, sometimes dark, which has the appearance of a nucleus. It is to the aggregation of the red corpuscles that the blood owes its red hue, although when examined by transmitted light, their color appears to be only a faintly reddish yellow. The corpuscles vary slightly in size even in the same drop of blood, but the average diameter is about 7.5 microns, and the thickness about 2 microns. Footnote: a micromillimeter, or micron, is 1/1000th of a millimeter, or 1/25,000th of an inch. End note. Besides these, there are found certain smaller corpuscles of about 1/2 of the size just indicated. These are termed microcytes, and are very scarce in normal blood. In disease conditions, for example anemia, however, they are more numerous. The number of red corpuscles in the blood is enormous. Between 4 million and 5 million are contained in a cubic millimeter. Power states that the red corpuscles of an adult would present an aggregate surface of about 3,000 square yards. If the web of a living frog's foot be spread out and examined under the microscope, the blood is seen to flow in a continuous stream through the vessels, and the corpuscles show no tendency to adhere to each other or to the wall of the vessel. Doubtless the same is the case in the human body, but when human blood is drawn and examined on a slide without reagents, the corpuscles tend to collect into heaps, like rouleaux of coins. It has been suggested that this phenomenon may be explained by alteration in surface tension. During life, the red corpuscles may be seen to change their shape under pressure so as to adapt themselves, to some extent, to the size of the vessel. They are, however, highly elastic, and speedily recover their shape when the pressure is removed. They are readily influenced by the medium in which they are placed. In water they swell up, lose their shape, and become globular, endosmosis. Subsequently, the hemoglobin is dissolved out, and the envelope can barely be distinguished as a faint circular outline. Solutions of salt or sugar, denser than the plasma, give them a stellate or crenated appearance, exosmosis. But the usual shape may be restored by diluting the solution to the same tonicity as the plasma. The crenated outline may be produced as the first effect of the passage of an electric shock. Subsequently, if sufficiently strong, the shock ruptures the envelope. A solution of salt, isotonic with the plasma, merely separates the blood corpuscles mechanically without changing their shape. Two views are held with regard to the structure of the erythrocytes. The older view, that of Rolette, supposes that the corpuscle consists of a sponge work or stroma permeated by a solution of hemoglobin. Schaefer, on the other hand, believes that the hemoglobin solution is contained within an envelope or membrane, and the facts stated above with regard to the osmotic behavior of the erythrocyte support this belief. The envelope consists mainly of lecithin, cholesterin, and nucleoprotein. The colorless corpuscles or leukocytes are of various sizes, some no larger, others smaller than the red corpuscles. In human blood, however, the majority are rather larger than the red corpuscles, and measure about 10 microns in diameter. On the average, from 7,000 to 12,000 leukocytes are found in each cubic millimeter of blood. They consist of minute masses of nucleated protoplasm, and exhibit several varieties, which are differentiated from each other chiefly by the occurrence or non-occurrence of granules in their protoplasm, and by the staining reactions of these granules when present. The most numerous, 60%, and important are irregular in shape, possessed of the power of amoeboid movement, and are characterized by nuclei which often consist of 2 or 3 parts, multipartite, connected together by fine threads of chromatin. The protoplasm is clear and contains a number of very fine granules, which stain with acid dyes such as eosin or with neutral dyes, and are therefore called oxyphil or neutrophil. These cells are termed the polymorphonuclear leukocytes. 2. A second variety comprises from 1 to 4% of the leukocytes. They are larger than the previous kind and are made up of coarsely granular protoplasm, the granules being highly refractile and grouped around single nuclei of horseshoe shape. The granules stain deeply with eosin, and the cells are therefore often termed eosinophil corpuscles. 3. The third variety is called the hyaline cell or macrocyte. This is usually about the same size as the eosinophil cell, and when at rest is spherical in shape and contains a single round or oval nucleus. The protoplasm is free from granules, but it is not quite transparent, having the appearance of ground glass. 4. The fourth kind of colorless corpuscle is designated the lymphocyte, because it is identical with the cell derived from the lymph glands or other lymphoid tissue. It is the smallest of the leukocytes and consists chiefly of a spheroidal nucleus with a very little surrounding protoplasm of a homogeneous nature. It is regarded as the immature form of the hyaline cell. The third and fourth varieties together constitute from 20 to 30% of the colorless corpuscles, but of these 2 varieties the lymphocytes are by far the more numerous. Leukocytes having in their protoplasm granules which stain with basic dyes, basophil, have been described as occurring in human blood, but they are rarely found except in disease. The colorless corpuscles are very various in shape in living blood, because many of them have the power of constantly changing their form by protruding finger-shaped or filamentous processes of their substance, by which they move and take up granules from the surrounding medium. In locomotion, the corpuscle pushes out a process of its substance, a pseudopodium as it is called, and then shifts the rest of the body into it. In the same way, when any granule or particle comes in its way, the corpuscle wraps a pseudopodium around it, and then withdraws the pseudopodium with the contained particle into its own substance. By means of these amoeboid properties, the cells have the power of wandering or emigrating from the blood vessels by penetrating their walls, and thus finding their way into the extravascular spaces. A chemical investigation of the protoplasm of the leukocytes shows the presence of nucleoprotein and of globulin. The occurrence of small amounts of fat, lecithin, and glycogen may also be demonstrated. The blood platelets are discoid or irregularly shaped, colorless, refractile bodies, much smaller than the red corpuscles. Each contains a central chromatin mass resembling a nucleus. Blood platelets possess the power of amoeboid movement. When blood is shed, they rapidly disintegrate and form granular masses, setting free prothrombin and the substance called by Howell thromboplastin. It is doubtful whether they exist normally in circulating blood. End of section 2.